All right, so first of all, I'd like to acknowledge the traditional owners on the lands on which we're meeting, and I pay my respects to their elders past, present and emerging. So I don't really have to do much of an introduction because Beck's already done a great job, but we've been really interested in developing double-stranded RNA as a control for myrtle rust. And so it's actually been developed for other plant pathogens. So it was first developed against viruses, and I've shown a virus example here. If I can, oh yeah, cool. So basically what we do is we synthesize double-stranded RNA that's exactly homologous to an essential gene in the pathogen that we're targeting. In this case, it's a virus. We spray it onto plants, and for the virus ones, they actually get taken up by the plants, and it triggers a natural defense mechanism known as RNA interference, which is naturally present, protecting plants from viruses. But it's also present in other eukaryotes, such as fungi, to protect against viruses, and also transposons and other sort of things in the genome. So what happens is that this double-stranded RNA, it gets recognized by dicer proteins. They dice it up into small interfering RNA, which then is sort of used to, it gets bound to other proteins, and it's used as a template to find any homologous RNA. So in this case, it's finding homologous virus RNA, but it could be fungal messenger RNA if we're targeting fungi. And we've seen that this can actually protect plants against viruses. And the beauty of this technology is that it's highly specific. So we can choose the sequence that will only target the pathogen we're interested in. It won't target any beneficial organisms. It doesn't leave any residues on the plants. We don't need to incorporate anything into the genome of the plant, so it's non-GM. And we can redesign our RNA very easily and redeploy if required. So these are some of the other examples in the literature where this technology has been used. So it's been used against insects, and a good example is the Colorado potato beetle. It's also been used against other fungal pathogens, so Sclerotinia, Fusarium graminearum, and Botrytis cinerea. And so basically you see a reduction in lesions on the leaves or on the fruit and the vegetables. So as Beck mentioned, we've recently published our work on RNAi, or double-stranded RNA targeting Ostropaccinia acidii. And this was a nice collaboration, as she mentioned, between a number of different groups. And so just briefly, an overview of our main findings. We found that the uridinospores could take up double-stranded RNA from the environment, and when we targeted specific essential fungal genes, we were able to reduce the germination of the uridinospores, and when they did germinate, they didn't form the typical infection structures, so they sort of started forming what looked like infection structures, but it didn't go further. And then this translated onto plants to disease protection. So what we're now interested in looking at is whether our double-stranded RNA can actually provide more of a systemic protection. In our initial work, we were just co-applying double-stranded RNA with the fungal spores, or applying RNA onto leaves and then inoculating those same leaves with the pathogen. So we wanted to see whether, first of all, whether the plants would take up double-stranded RNA, and once the RNA was in the plants, whether it would actually move to other parts of the plant and then protect different leaves. So Chris Brosnan at UQ did a lot of pioneering work in this area. This has been published in bioRxiv, and he's just resubmitted this. But basically so he did a lot of work in Arabidopsis and other sort of model crops. So what he did is he actually applied double-stranded RNA to Arabidopsis leaves, And then he sampled from distal tissues, so from flowers and from roots. Sorry, it's a bit hard to see the screen from here. But basically, he could detect the double-stranded RNA in those distal tissues 24 hours later. And in the roots, we actually did some earlier time points. So we actually saw the double-stranded RNA move to the roots within an hour. So it seems to move very quickly, at least in this very small plant. And it's sort of maintained as well. So in this experiment, when he applied the double-stranded RNA, the plant wasn't flowering. But then he sampled 2 weeks later, and he actually detected that double-stranded RNA in the flowers. So it's sort of being maintained in the plant. These are healthy, non-infected plants. But then we went one step further and sort of looked at, well, will the RNA actually protect against a pathogen, or not protect, but at least knock down a targeted pathogen gene? In this case, we were using GFP-expressing fungi, so Verticillium dahlia and Fusarium oxysporum. And when the RNA was applied to the leaves, it actually did knock down root fungal pathogen genes. So that was the GFP gene we were looking at. So that shows there is some promise to get systemic protection. We haven't shown protection yet, but that's kind of the next thing we're looking at. So I was interested in whether we could use any of these concepts for the Myrtaceae species we're interested in. So first of all, I was just curious, well, do any of these Myrtaceae take up double-stranded RNA? So I just sprayed some different species with RNA, so Syzygium jambos, which is the rose apple that we were doing a lot of our original work on, as well as lemon myrtle. And so basically what I found was the RNA was still present on the sprayed leaves 1 week later, but it wasn't present in any of the new growth, so the leaves that hadn't had any RNA applied to them. So at least it wasn't moving in the sort of time frame we'd seen for those other plants. And we tried spraying both sides of the leaf, so the lower surface and the upper surface, and we still didn't see any movement. And this was also with a penetrant, because we thought, well, we'll try and help the RNA get into the leaves, but we still didn't see anything. And we also looked at young leaves in case it was something to do with the leaf age, but didn't really have any success. So I was reading this paper, which was quite interesting. They were actually delivering RNA into woody species, and they were using trunk injections and pedial absorption. So these are some of the results from that paper. So they were looking at apple trees and grapevines and doing some trunk injections, and they were able to detect RNA in the new growth at sort of early time points within a few days. And then there was this other method where they were doing pedial absorption, where they're actually soaking the pediols in the double-stranded RNA and then detecting the RNA in the leaves. And this is just showing in tobacco. So I decided to do this. I'm also working on Phytophthora cinnamomi. So I had some avocado plants, and we were sort of interested in whether we could spray RNA onto the leaves and if it would go into the roots to protect against this root pathogen. So this was all done with Louise Shuey at DAF. And so what we did is we actually injected some double-stranded RNA into these avocado trees using the trunk injection approach. And then I sampled at different time points afterwards. And then I sampled leaves and roots. So this northern blot is 6 weeks after I did that initial trunk injection. And so I was actually quite surprised, but I could still see the double-stranded RNA in the leaves and in the roots at that time point. And I also sprayed the RNA onto leaves as well. And it seems that avocado can actually take up RNA really well. So even when I sprayed it onto the leaves, I could still detect it in the roots. So yeah, avocado seems to be a bit different to some of the other Myrtaceae I was working with. But then, if it will go. So then I tried some PDL soaking in some different Myrtaceae just because they're a bit small, so I couldn't really do the trunk injection. So I just decided to soak PDLs in the double-stranded RNA. And so then I took samples from the new leaves to see whether there was any RNA present. And this was at 1 week after I applied it. And so it looks like it might be a bit degraded, but there was double-stranded RNA present in the new leaves from a few different species of Myrtaceae. So that is kind of promising. It shows that we can get RNA into these plants, and the next step is actually doing a protection assay. So we'll try infecting some of these plants and see whether RNA, when it's inside the plant, can protect against myrtle rust in the same way as when it's sprayed onto the plant and is present on the leaf surface. So yeah, just some other questions. We're also looking at the native plants. We're really interested in whether we can protect them. Obviously, they are the ones at risk of becoming endangered. We want to move into a field environment, see whether this holds up in the field. All of our work so far has just been in the glasshouse. So we want to see whether we can also achieve protection under field conditions. And another question that we're looking into at the moment is, why is this working so well in Osteopuccinia acidii? Because we are looking at other fungal pathogens, but double-stranded RNA seems to be particularly effective against Osteopuccinia. So we're sort of really interested in understanding why that might be. So I'd just like to thank everyone who's involved in this work. So Beck, Louise Shuey, Alistair McTarget, a lot of different people from UQ, and our collaborators and some of my industry partners. Thank you.
Thank you so much. Any questions for Anne after that talk? Yes, Mia.
Hello. It might be a basic question, but you mentioned when it's applied by spray that it doesn't appear on the new leaves. So is the response that you're looking for, the immune response of the plant, systemic, and therefore, are those new leaves still protected while you're detecting it on the old growth, or are the new leaves then vulnerable because the effectiveness is leaf specific, like you need to coat every leaf for the thing to work?
Yeah, so the whole process, the processing of the double-stranded RNA looks like it's happening in the fungus, so not in the plant. So the reason why we think the RNA is so stable in these plants is it's not getting processed at all. So it's not going into cells. It has to go into a cell for the RNAi machinery to act on it. So it appears to just be floating around in the vasculature of the plant, but then when the pathogen does infect, it actually gets transferred into the pathogen and processed by the pathogen's RNAi machinery. Yeah, so yeah, I think that answers your question.
Yeah, thank you.
Just one more.
I had two questions, actually, based on one of those followed from for the question is, so my understanding is when you applied these double-stranded RNAs, they were actually targeted towards the fungus, and you tested the uptake. But have you actually tested the uptake of double-stranded RNA, which actually has a target in the plant, and you maybe be able to see a signal better if the plant machinery actually kicks in and amplifies your double-stranded RNA?
So we haven't really had much luck getting RNA, like endogenous plant genes downregulated with RNA. It's quite tricky, but the best success has been with the viruses, I guess, and that's sort of using the plant's machinery to target the virus. Yeah, but targeting endogenous genes has been really tricky, and there's not much evidence of it in the literature either. Yeah.
Can I ask another one? I had another one, which was, so at the end you pointed towards that this double-stranded RNA works really well in apsidiae, but not some of the other rusts. Do you have any indications this is an uptake of the double-stranded RNA in the other rusts or in the machinery which works differently in the other rusts?
Yeah, we're still looking into what it could be, but definitely uptake could be something because we have looked at RNA uptake into a lot of different fungal pathogens, and it definitely gets taken up really well by Osteopuccinia acidiae and not so well by some other pathogens. So uptake could definitely be part of it. But we are also doing some small RNA sequencing to look at the actual processing of the RNA and comparing that between different fungal species just to see what's happening on that level as well.